Welcome back to the deep dive, everyone. Today we're taking a look at something pretty incredible. It's all about proteins, those tiny machines that are essential to life. They do everything, build our cells, fight off infections, and much, much more. But have you ever wondered how they actually do it?
It really is remarkable, isn't it?
It is. It all comes down to their shape, their three-dimensional structure. And what's really mind-blowing is that scientists are now learning to redesign those shapes, like actually engineer proteins to give them totally new capabilities.
It's a field called protein engineering, and it's rapidly advancing.
We've got some really interesting research articles here that delve into this world.
Yeah, they provide a great overview of the techniques and principles involved.
So our mission today is to unpack those core strategies, help you understand how scientists are redesigning and even creating proteins from scratch, without getting bogged down in the super technical details, of course.
Right, the goal is to give you a clear picture of this cutting-edge field.
So one of the key takeaways from the research is that there are three main approaches to protein engineering: rational design, directed evolution, and semi-rational design. Let's start with rational design because it kind of lays the foundation. The core idea here is that if you know enough about a protein's structure and its mechanism, like how it works at the molecular level, you can make very specific changes to its amino acid sequence. Those are its building blocks to achieve, a desired outcome.
Yeah, and what's amazing is the level of detail they can get. They use techniques like X-ray crystallography. You can think of it like taking a three-dimensional photograph of a protein and cryo-EM, which is a really powerful type of microscopy. And now we even have AI predictions like AlphaFold to help model protein structures.
I've heard about AlphaFold. It's really revolutionized the field, right?
Absolutely. And once they have a good understanding of the structure, they use computer simulations, things like molecular dynamic simulations and quantum mechanical calculations, to predict how changes to the amino acid sequence will affect the protein's behavior.
So it's like having a virtual lab where you can test out different modifications before you actually go and make them.
That's so cool. And the research talks about some specific techniques that fall under rational design.
Hmm.
One that really caught my eye was site-directed mutagenesis. They call it SDM for short. It sounds super complicated, but it's basically a way to change a specific amino acid in a protein's genetic code. They use methods like PCR, which is a way to copy DNA, and even CRISPR, that gene editing tool we hear so much about. It's like having molecular scissors and a very precise instruction manual.
Yeah, you can think of it that way. And if you think about the applications, it's really powerful. For example, in drug development, you could use SDM to design a drug that fits its target protein perfectly, like a key in a lock. This could make the drug more effective and reduce side effects. That's amazing. And then there's domain shuffling. That's where you take functional parts, like domains, from different proteins and you combine them, almost like mixing and matching, to create a totally new protein with potentially brand new functions. It's like taking different tools from a toolbox and putting them together to build something completely novel.
It's a bit like that, yes. And the possibilities are vast.
Okay, this next one sounds like something straight out of science fiction. De novo protein design. That's literally designing proteins from scratch, not just tweaking existing ones.
It's one of the most exciting areas in protein engineering, and it's becoming more and more feasible thanks to advances in our understanding of protein folding and the development of really sophisticated computational tools.
The research mentions this program called Rosetta, which uses algorithms to predict how a sequence of amino acids will fold into a 3D structure. And again, deep learning is playing a big role here, right?
Absolutely. Deep learning algorithms are helping to improve the accuracy of these predictions and allowing scientists to design proteins with increasingly complex functions. Imagine designing a protein that can self-assemble into a nanoscale sensor. That's the level of potential we're talking about.
That is mind-blowing. So that was rational design. Now let's switch gears to directed evolution. This is a totally different approach. Instead of meticulously planning every change, it's more about letting nature do the work.
It's kind of like breeding dogs, but at the molecular level.
Huh. Yeah, I can see that. So you start with a gene that codes for a protein and you introduce random mutations into it. It's like creating a huge library of slightly different versions of the protein. Then you screen those variants to see which ones have the properties you're looking for.
And the research highlights some of the key techniques they use to create that initial diversity. One is error-prone PCR. Basically, you're using PCR, but you would intentionally make it sloppy so it introduces random errors mutations into the DNA sequence.
So it's like a genetic photocopier that makes mistakes on purpose.
Exactly. And another technique is DNA shuffling. That's where you take fragments of DNA from similar genes and you shuffle them together, like a deck of cards, to create new combinations.
Okay, so you've got this huge pool of protein variants, but how do you actually find the ones that are the best?
That's where things get really clever. The research talks about these techniques called display technologies. One example is phage display. They actually display the protein variants on the surface of viruses called bacteriophages. Then, they use a target molecule to fish out the phages that have the protein variant that binds most tightly to the target.
Oh, wow. So it's like a molecular fishing expedition.
Pretty much. And there are other display technologies like yeast display, which uses yeast cells, and ribosome display, which is a cell-free system. These are incredibly powerful tools for screening and selecting proteins with specific properties.
Okay, so we've got rational design and directed evolution. Now let's talk about the hybrid approach, semi-rational design. So it's not as knowledge-driven as rational design, but it's not completely random like directed evolution, right?
Exactly. It sits right in the middle. The idea is to use structural insights to identify key regions of the protein, they call them hotspots, where changes are most likely to have a big impact on the desired property.
So instead of making changes everywhere, you're focusing on those specific areas.
Right. And then they use targeted mutagenesis, which is like site-directed mutagenesis, but more focused, to introduce mutations specifically in those hotspots.
It sounds like a really smart strategy, a way to streamline the process.
It is. It reduces the search space and makes it much more efficient.
So we've covered the main approaches, but the research also highlights some important considerations for protein engineers. Protein folding and stability is a big one, right?
Absolutely. When you change the amino acid sequence, you can disrupt the delicate balance of forces that hold the protein in its correct three-dimensional shape.
So it might not fold properly, or it might be more prone to falling apart.
Exactly. And that can obviously affect its function. So protein engineers have to take that into account. They use computational tools to help predict the effects of mutations on folding and stability.
And what about protein dynamics? The fact that proteins aren't static, they move.
That's another crucial factor. Protein movement can be really important for its function. It might be involved in binding to other molecules, catalyzing reactions, or transmitting signals. And just like with folding, mutations can affect protein dynamics.
So computer simulations come in handy here too.
Definitely. They can help us understand and even engineer protein movements. And really underpinning all of this is computational protein design. It's using computers and AI to design, analyze, and predict how proteins will behave. It's a rapidly growing field that's making huge contributions to protein engineering.
It's amazing how much computational power is involved in this field. So to sum up, we've explored the main strategies for engineering proteins: rational design, directed evolution, and semi-rational design. And the key principle is that a protein's three-dimensional shape dictates its function. By changing the amino acid sequence, scientists can essentially reprogram these molecular machines to give them new and improved abilities.
And this has huge implications for all sorts of fields. We're talking about creating new therapies for diseases, developing more efficient enzymes for biotechnology, and even designing new materials with unique properties.
It really is a revolutionary field. So here's something to think about as we wrap up. As our ability to design proteins from scratch gets better and better, what are some of the most transformative possibilities you can imagine for the future of technology and healthcare? Food for thought. Thanks for joining us for another deep dive.
It's been a pleasure.